Welcome, everyone. It's my pleasure to introduce you to our today's speaker, Juan Balbuena, who comes from Valencia, from the Instituto Cavanilles de Biodiversitat y Biologia Evolutiva. He is professor there at the group of computational biology. Sounds very interesting. He studied in Valencia, did his PhD there as well, and a few years after his PhD defense, he became professor titular of that university, so his career has been linked to that university. It's interesting that he did his stay abroad after getting a professorship. So he did his experience abroad with European funding in '98. He went to Norway, to Bergen. He has had, ever since, quite some European funding of different natures. He is also evaluator of European grants. Young people take note, and other people. So he works on coevolutionary relationships between symbionts, parasites, and hosts. He does most of his work on fish, marine fish. And a lot of his work is very theoretical. He has developed methods for doing this coevolutionary analysis. He has written packages, analytical packages. He has several of them published in systematic biology, but he also has developed some theoretical grounds, and he has some of that stuff published in Tree. As I told you, he has had several European projects working on some applied stuff on marine fish, some stuff on cod and gray mullet, but also in aquaculture. So he has a quite broad scope of interests, and I leave you with this.
Thank you, Marta. I'm really thrilled to be here today, and very grateful and honored to be in the National Museum of Natural Science to deliver this talk to you. I will start. Marta has spoken a little bit about myself, but I'm going to introduce a big part of our research line in Valencia. And to justify why we took this way, I have to do a little bit of history. So you see that my PhD was long ago, and logarithmic scale is not too far from the Devonic. Then I specialize in marine parasitology in general. And I always like to have some numerical taste or do some applications of numerical methods, biometry, to my studies. As Marta has said, I have led a number of European projects, and actually at that moment in time, about 10 years ago, more than 10 years ago, I wanted to follow a new course in my career, because I was losing touch with research. I was merged in a lot of bureaucracy, managing projects, and I was really unhappy at that time. So I decided to downscale somehow, and at the same time get to more contact with, I wanted to do my own research apart from supervising others to do the research. So I decided I was looking for a new area to work with. And this area, in a way, had to be based with a few ideas, like since my PhD was on whale parasites, immediately one idea was how these parasites came to colonize these whales historically and ecologically. So there was an idea there that is really attractive. And sometime back in time in the 80s, I learned some programming, and I discovered actually that the programs I used at that time was like dead languages, right? So I have to look for new things. But anyway, I had this in mind, and looking, and one of the researchers I admire most is Pierre Legendre. And reading one of his papers published in 2002, which is the description of a test called parafit, I thought, okay, that's something that I could develop, something that I could work with. And so I had this idea, and it fulfilled the first condition to start a new research line in the last leg of my scientific career. And the first condition was that doing this sort of thing is cheap. You don't need a big lab, you don't need expensive equipment, you just need a good computer and ideas, and so I like it. So, but at the same time, I thought, okay, it has to be relevant at the same time, and I have to find something that is exciting. So it's looking at test coevolution between symbiotic partners is relevant? Well, I think yes, because a key question in biology is to study how species interact. And here in this slide, I wanted to represent how the species interact in the present, that is ecologically in time, but also along the temporal scale. Probably these interactions change in time, how they evolve. They can explain what we see today. So this always has been a central idea in biology ever since George Darwin, when he talked in The Origin of a Species about the tangled bank, when he realized that the species interacted between them in very special ways. Still, Darwin didn't have a very clear idea about things like evolution or something like that. It was understandable because it was very early, so there were very similar ideas that needed to be developed along the centuries. Preparing this talk, I found a lot of interesting stuff to really explain how the ideas like coevolution, symbiosis came to be. But it would be a really long talk, so we will just jump in time, make a big leap in time. But anyway, he in his next book that published 3 years after The Origin of the Species, that was the first thing he published. He spent 3 years without publishing anything. So he published this book about the British orchids, and he was British orchids and other orchids. And he was really intrigued by Madagascar orchid with a very long nectary. And he predicted that there should be a moth or some insect able to use to pollinize this orchid. And actually, some century later, the moth was found. And recently, I think it was in 2005, it was recorded on video how this moth was pollinizing the orchid. So in a way, what this is telling us is that there is a predictive power in Darwin's idea and the idea of coevolution and so on. And this has been also reinterpreted later by some authors like a coevolutionary race, even though I think Darwin didn't have a very clear idea about what coevolution was. But anyway, was something there, something starting. And as I said, we have to jump 150 years to today, to these guys, Charleston and Libeskind had us in this book. They paraphrase the famous sentence that nothing in biology makes sense except in evolution. So you can turn this around and say that nothing in evolution makes sense except in the light of coevolution. So I have illustrated this concept here with the famous pollinizers of fig trees. That this is fascinating, actually. It's very complicated. You have pollinators, you have predators, you have parasitoids. Well, you name it, is incredible. And also, since I'm a parasitologist, I found this very classic example of the scolex of a cestode that really fits the intestinal surface of ray fishes. Well, anyway, we say, yeah, nothing in evolution makes sense except in the light of coevolution, and this is especially true when we go to symbiotic relationships that we can describe vaguely as mutualism, commensalism, parasitism. I suppose you are familiar with these terms. I'm not going to extend about that. The only thing I wanted to say is that I made this overlap to indicate that the borders are somehow blurred, but well, symbiosis, two organisms that live together and somehow they have a kind of very tight relationship. And we find that this symbiosis is really essential to explain many things in biology. We are realizing this. Preparing this talk, I just found this paper from this year where they were looking at insects, and they were comparing insects with obligate and non-obligate intestinal symbionts. And you can see here that species richness is very much dependent on having or not obligate relationship with the symbionts. So if you overlook symbiosis in many organisms, you may be overlooking a very important driver that explains, for instance, diversification, that explains many processes, many evolutionary processes, right? Well, so yeah, it is relevant. And the third idea is I'm going to work with this thing for the next 10 years or so. So it has to be really exciting. So this is the next thing that we are going to talk about. And in fact, one of the things we do in the lab is to study patterns of macroevolutionary change, more or less. How you see, what you do is compare phylogenetic trees and how these phylogenetic terminals relate to each other. Here you see that the first studies on these sort of things were they were very old, and they started with host parasite systems. And there is a very famous rule that is the Fahrenholz rule. That is more than 100 years old. That what is really saying in a way is what we could say today is like the parasites. What they do is they do a phylogenetic tracking of their host. So they follow closely to Fahrenholz. They will follow exactly the diversification of the host. You can, of course, reinterpret this because it's not always the case. For instance, here, how these pubic lice which is related to gorillas finishing in humans is a bit embarrassing. But if you compare the two graphs, you can see that the phylogenies, they match very closely, not only in terms of topology, but also in terms, for instance, of the nodes ages are very, very close. And this indicates more or less, or in a way, reinforce the Fahrenholz rules in saying that parasites follow the phylogeny of their host. But obviously in Fahrenholz times, there were no idea to estimate phylogenies in an independent way. So we have to wait until the 1980s when we started with PCR, structure, sequencing. So we started to be able to build phylogenies of one partner, one symbiotic partner, and other symbiotic partner independently. And we come to this very early and seminal work of Hafner and Neidler of 1988, where they compare the phylogenies of pocket gophers and their chewing lice, which is a very famous work. They published 3-4 papers about this relationship. That in a way, they more or less compare and came to the conclusion that actually there was quite a congruency, the phylogeny of the two organisms. By the way, I just take the chance to explain that what we see here is usually called a tanglegram. That is, you have the two phylogenies of the organisms you want to study, and then you also indicate in the diagram, you indicate the relationships. So that means that this parasite species is related, is found only in this host species, and so on. So you have, in a glimpse, you have both the evolutionary history of both partners and also how they interact in ecological time. Okay, but as soon as we started to build phylogenies and so on, we found that, well, guys, the Fahrenheit rules may hold sometimes, but is very far from a general rule. Here you have the relationship between mammals and fleas. You can see this is really chaotic, this is really messy. So there is no Fahrenheit rule, guys. In these situations, when they're so complex and so on, I just came with the idea that this looked like the evolutionary Gordian knots. No? If you are familiar with the history of Alexander the Great, there was a knot, and the one able to disentangle the knot would be the king of, or the sovereign of Asia. And well, I'm doing a spoiler because I come later to this idea. Well, I call it a Gordian knot. Okay, why we find this Gordian knot? Because basically because there's not that simple. Because parasites get associated to hosts in two very different ways. So in a way, we have the descent, that is, they have been associated evolutionary with the host for a very long time. I have placed here Enterobius vermicularis, which is the pinworm, I think it's called in English, very typical in children and so on, that we know that relatives are present in primates, so you more or less guess that they are here because of this historical association. But at the same time, parasites like host switching a lot. Well, I wouldn't say but they're always trying. They're always exploring the idea of colonizing a new host. So they're very good at that. Okay, host switching is not very frequent in ecological time, but it is in evolutionary time. So give them time enough, and give them possibilities to encounter repeatedly, and it will for sure happen. So here I have a place, because we as humans, we like water, we like playing in water. This is a very human thing. And so we are exposed to aquatic environments where we can get new parasites like schistosomas. Why we got schistosomas? I would say because we go to places where there are water. Okay, so we have two whole different processes, and these two whole processes are intertwined in evolutionary history. So that looks very interesting. Okay, now here I have in this slide, I have more or less divided everything that can happen with a host and a parasite. Okay, all the possibilities. And you can see that this is quite complex. I'm not going to go through the whole slide, but basically you have processes that have to do more with this phylogenetic association. The most simple one, of course, is co-speciation. So the host speciates, the parasite follows the host or speciates at the same time. There are other possibilities. For instance, a parasite can duplicate and form 2 species on a single host, et cetera, et cetera. Okay, so all these processes, what they do is they contribute to entangle the evolutionary Gordian knot. Okay, and this is especially extreme in cases of host switching, where here these parallel lines what indicate is that the hosts are not phylogenetically related. They are not very related phylogenetically. So you have different possibilities. For instance, the parasite can jump to a new host and maintain some genetic flow between the 2 hosts. They can speciate. Okay, many things can happen, but all this contributes to really entangle this Gordian knot. Therefore, I had my must-be list complete. Let's start a research program in cophylogeny, and let's see more or less how it works. So cophylogeny, as you say, its same is explaining these shared evolutionary histories. And initially, the 2 main ideas basically are if the evolutionary histories are congruent, so the idea is to compare the 2 phylogenies, and also to explain which evolutionary events, I mean, by event, well, you will see it later, okay? Basically, we have 2 approaches, okay? So we have the global fit approaches, we have event-based methods. Global fit approaches, they attempt to answer this first question that biologically can be translated as, was co-speciation important at all in this particular system we are looking at? Okay, so the idea of congruence, initially, I know this is controversial, but the idea of phylogenetical congruence means, or in a way assumes, that co-speciation was an important driver in the system we are studying. Okay, whereas event-based methods attempt to really reconstruct the events. So taking into account these events I presented to you before, which events were involved in the system we are studying. Well, let's start with global fit approaches. As said, they assess phylogenetic congruence. Later in the literature appeared the term coephylogenetic signal. That sounds very cool in a way, but by now it's just a new term and it's not really well defined. I suspect that the idea of talking about coephylogenetic signal instead of phylogenetic congruence has to do with the will to quantify, to give a quantifying twist to the test, okay? But let's treat phylogenetic congruence and coephylogenetic signal like more or less synonym by now. And so the idea of coephylogenetic signal is if you have really congruence between the 2 histories, you say there's high coephylogenetic signal. If there is no relationship, you can say that the coephylogenetic signal is low, okay? And so we come back to the famous paper by Legendre et al., where they developed parafit. This was a very interesting paper to me that inspired me to really do an alternative to parafit, to find something additional that I think would give a more graphic display of the analysis. But basically, all global fit approaches are based on somehow comparing distances matrix of the host and the symbiont. So essentially, if you have a phylogeny, you can reduce the phylogeny to a distant matrix that can be a patristic distance, a genetic distance, whatever, but it can be converted into this sort of data. And Legendre used or converted, translated this distance matrix to an Euclidean space by means of principal component analysis. So essentially, you can represent a phylogeny into a multi-dimensional space. So it's a kind of attractive. Phylogenies are not Euclidean, so something can be lost in the translation to Euclidean space, but it's a very handy way of incorporating phylogenetic information into a statistical framework. So you can do statistics with this configuration, okay? I'm not going to explain in detail part of it, but it was based on the fourth corner problem. That is a problem that was used in ecology. So that simply shows that you can take methods from other areas and adapt them to new areas, correct? So basically what they do is they use these principal component coordinates of the both phylogenies, and they use the matrix of associations. So they make a matrix where zeros represent no associations and ones represent associations. I'll show you this later. And compute a statistic that tells you about the strength of association between the two partners you're studying, correct? Okay, so that gave me the idea that using the same sort of data, it could be used in a Procrustes way. So I developed what was called Procrustes approach to cophylogeny. The acronym is PACO, in which we treated phylogeny like a kind of biological structure, okay? So what we basically did is we have a hyperspatial configuration that is this phylogeny. We have a hyperspatial configuration that is this other phylogeny, and we found a way to do a Procrustes superimposition to see how the two configurations fit together, okay? So the algorithm is explained here, is in the paper, but basically you have the phylogenies, distant matrices, you convert the matrices into the principal coordinates, and then you use this matrix that I said before, which is the matrix of association between the partners. So here it encapsulates the information in these lines of association, okay? So then we did some trick, because to do a Procrustes superimposition, you need the same number of points to superimpose in space. So what we did is, when we had a host that was associated with two symbionts, we duplicated this row in the matrix. And when we had a symbiont that was associated with two hosts, we duplicated the column in the matrix. And then we duplicated the rows in the principal coordinates. And so voila, we had the two configurations, and we were able to superimpose them. To simplify the idea, this is presented in 2 dimensions, but of course it's in n dimensions. But more or less what the basic idea is that you can see how well the two configurations fit, and you compute this would be the residuals, and the smaller the residuals, the two phylogenies, will be closer together, will be more similar, or the general pattern of associations, and phylogenies would be more similar. Therefore, you sum the square of all residuals, and you can do some test to say, for instance, if this sum of square residuals is larger than what you would expect by chance. Okay, this method was later developed as a package in R. But basically, it's more or less what I was anticipating to you. You have the sum of square residuals, so you sum all the, again, this is in 2 dimensions, so you have to do it in the n dimensions, but anyway, you sum these quantities, you have the total amount. This is what you observed, for instance, in this particular example that I presented to you before, sorry. You have, this is the m square found, so you do 1,000 and 1,000 simulations, and you can see whether it's in the 0.05 percentile or not, so it is outside. Therefore, you can say, you can give a p-value saying that these associations did not happen by chance, so it's very unlikely that these associations happened by chance, basically is the idea. And then you can also look at the individual residuals. Each residual is associated to one of these associations, so you can conclude which associations, for instance, are the most incongruent in a given Cophylogenic or in a given targetgram. So we can explain. So this is more or less what is here. Parafit also can do the same. So basically, this is what Paco produces, this kind of bar plot. And the results were compared with Parafit from a paper. But basically, you can identify, for instance, these 4 or 5 associations. They are really incongruent, at least compared to the rest of host-parasite associations. Okay. How long can I talk? Okay. I'm talking about global fit methods. They have, of course, pros and cons. You have here a few. They handle very large data sets. They provide a p-value for people that like p-values. You can do the same thing, this very same thing of looking at the individual residuals. If you have, for instance, posterior probability trees in a Bayesian reconstruction, so you can use these 1,000, 2,000 trees, pairs of trees, to do exactly the same, and therefore you can compute confidence intervals for each of the associations. So in a way, you can make a more informed decision about the nature of a particular residual. For instance, here in this case, you can see that it's very high. You could say that it's very incongruent, but the confidence interval is overlapping the median value. So okay, but not really conclusive. Whereas these two, for instance, they are really high, and the confidence intervals do not overlap the median value of residuals. So you can pick up these two as especially incongruent. Okay. Another quality of this type of methods, and particularly of Paco, is that they have proven very effectively in a quite different, I don't get the word, situation. It's not what the word I have, but in a quite different problem. That is when you have phylogenies built with different markers. So you can compare, in this case, for instance, you have plasmids, because I have 2 papers on orchids. I'm a zoologist, so I'm very proud of my 2 orchid papers. So here you have a nuclear phylogeny and an organellar that is a plasmid phylogeny. And you can compare the phylogenies and identify the positions that are incongruent. So I think it's very interesting when you are really building large scale, or you are doing large-scale phylogenetic work. This is not cophylogeny, but as I said before, you can take tools from different areas and apply them to other areas. Then, of course, there are cons of global fit methods. For instance, one thing we are realizing now in our lab is that they are very anti-conservative, both parafit, you name it, the R of Homola et al., Paco. They tend to give you a significant P with the least deviation of randomness. You have something that is not random in plot. Yeah, it's significant. Okay, so I think the null hypothesis of this test need to be rethought somehow. I don't want to scare you, but be careful with the significant p-values. Well, anyway, and this, as I said before, phylogenetic congruence is not necessarily indication of co-speciation. Then we have event-based methods that try to, as I said, explain what's happened. So basically, they try they assign costs to evolutionary events. So they try to explain. Here you have identical histories explained in a different way with different events. And what you do arbitrarily is you apply a cost for each event. For instance, duplication, you assume that the cost is two, Host switch, you assume that is 3, so you assume that host switch is more likely to happen in your system than duplication, for instance. On co-speciation, you assume that it's the easiest, so you assign a cost of 0. So you can try different combinations for the same history, and finally you pick up the combination that gives you the lowest cost. And so you have an explanation of what happened. For instance, in this case, you have first a co-speciation, then here you had a loss, here you have a host switch. So you give a full explanation of the whole history. We have also used this kind of analysis in our lab. For instance, in a study where we compare 3 monogenian genera, and surprisingly, we found that in the 3 genera and in general in other studies, in monogenians, which are parasites of gills of fish, the co-speciation is not really an important driver in the diversification of these organisms. So that makes you think. Event-based methods, they give you an explanation. Usually you feel very comfortable when you have history in your data. We are also doing some research, and they actually are very good at predicting the actual co-speciation events that happen in the history. But of course, they also have some cons that are expressed here. I'll not go into detail, but in conclusion, there is no perfect method. So you have 2 ways. You have global fit. You have event-based, used both in your studies. Then this is a twist of basically a global fit approach, which is the Alexandrian approach to the Gordian knot that consists basically in, as Alexander the Great did, he came to the temple and he just cut the knot in the middle and became the Lord of Asia. But basically, this is more or less the original algorithm. So basically what we do is we take a subsample. This is the whole tanglegram. So we take a subsample of the tanglegram. For instance, we take 5 associations of this tanglegram. Okay, so we run, for instance, Paco. We compute the M2, the sum of square residuals. And we do this 10,000 times. So you end up with a vector of 10,000 values, which is the values of the total sum of square residuals. And if you remember, when the square residuals are low, you say that there's high congruence between the two histories, when the square residuals are high, the congruence is low. So if we represent the distribution of, this would be the M2 values, the residuals values that we computed, and we focus on the area where the residual values are the lowest, we are expecting to find here the associations that are more represented in this area of the distribution, right? So basically, this is what we had. So for instance, this, for instance, would be very congruent, and this, for instance, could be a very incongruent link. Then we have to have some adjustment for, well, it would be very long to explain, so we compute some residuals. But in summary, this is what we did, and we have a distribution of residuals, and we computed a Gini value that, in a way, informs of the nature of the problem you are working with. Okay, so depending on this Gini inequality index, you can say, for instance, if it is very low, you have maximal co-speciation. When it is high, the value is very high, the co-speciation is expected to be low, basically. Okay, so we have, in addition to having a p-value, at least we have some statistic that informs on the strength of the relationship. Okay, then recently, this was the first chapter of my PhD student, Marc Javeria. It was published this year. So we converted random tapas into a R package with an additional algorithm that does essentially the same thing, but in a different way. Again, this method can be applied to pairs of Bayesian posterior distribution trees, to compute confidence intervals, to these frequencies of appearance in the distribution. Therefore, you can conclude, for instance, you can conclude here that this association is clearly incongruent because the confidence interval does not overlap, is different from 0, okay? Well, basically, this is, again, it's also a very graphical method because you can map the frequencies on your tanglegram. So you can identify the links that are more congruent in your history. And you can also compute some kind of fake ancestral states. You can use algorithms for estimation of ancestral states to see basically how the frequencies propagate down in the nodes of the phylogeny. So you can identify clades that could be more congruent than others, for instance. And this gives you information, for instance, in this case where we had the association between fleas and mammals. So you can identify clades where, at least in case of the mammal phylogeny, you can identify clades where congruence is higher and clades where congruence is very low. And was not the case in the case of the fleas. So it seems that the fleas were primarily associated with this large clade of rodents, and later they did host switch to other clades of rodents and other lagomorpha, for instance. Okay, so it's giving you a visual clue of things that could have happened in the past. Just to conclude, we see that we can somehow say, okay, cophylogenetic signal is high in a system, or cophylogenetic signal is low, but we need to go a step forward because we need to know which mechanisms could explain this high cophylogenetic signal or this low cophylogenetic signal. So you have here, for instance, if you find high cophylogenetic signal, it could be due to coevolution, to phylogenetic tracking, to vicariance. Which of the three is more likely to have work in this area? Or if cophylogenetic signal is low, it could be due to what is called diffuse coevolution. So we needed to give an additional step, so we came up with the idea of the Cophylogeny space that was inspired by a first paper of Althoff et al. Later, Russo et al. Developed a test that, in a way, served as a basis of our Cophylogeny space, in which we basically evaluate 3 dimensions. So we compute a value for cophylogenetic signal, a value for what we call host interaction. That could be the parasite tracks the phylogeny of the host, and another dimension in the space that is the symbiont interaction, that is the symbiont does the phylogenetic, no, the host does the phylogenetic tracking of the symbiont. I think I have explained it right, or maybe the opposite, but more or less this is the idea. Well, in practice, how we do this thing? Basically, what we do is we test the cophylogenetic signal using a method for cophylogenetic signal, and then we apply the same method to compare phylogenetic distances with trait distances of one partner, and in this case would be a parasite. Okay? So we are evaluating here the phylogenetic tracking, and here we compare the phylogenetic distance of the symbiont with the trait distances of the host. So we have 1 value, 2, 3, that is 1, 2, and 3 dimensions, and this point would represent the position in the Cophylogeny space of this particular problem we have here. So we expect in the future, when this method, if it's standardized, we could find some patterns in this Cophylogeny space in 20 years' time or something like that. So this is one of example, or recent example, of the application of the space. Here we computed a Procrustes R square, which gives you the strength, and actually the strengths were very low, so we compare basically the 2 phylogenies. Then we compare the phylogeny of the parasites with morphology of the fish. I know this is not exactly, but okay, this is what we have. Okay, and then we compare the phylogeny of the host with the morphology of the parasite. Basically, it was the ventral anchors and the dorsal anchors. These are anchors that they use to attach to the fish. So they have dorsal and ventral, so we had 2 comparisons instead of 1. And basically, what we find is that the largest was host interaction. Therefore, probably the parasites, and not surprisingly, parasites track the host rather than having a more coevolution or reciprocal relationship. Okay, it's more like following the host, which is quite logical, I would say. So finally, just to end some few ideas for the future. So first thing, we need to define better ways to measure this cophylogenetic signal if we want to, again, have some kind of a standard for the future. We also need perhaps to integrate information of the bipartite network. Hutchinson et al. Some years ago, they published a very interesting paper that has not been explored in its full, and I think could be very interesting, because in these networks you have modules, so you perhaps can examine these modules instead of the whole history. You can examine regionally, globally. So there are dimensions that still need to be explored. Another area of research could be to downscale the cophylogenetic problem, which is based on species, to individuals. And this is what we are doing currently in our project of the national plan, which is based on a very young host symbiont system. And I just would like to end the talk to giving a big thanks to very nice collaborators and very young collaborators that have a brilliant future, particularly Oscar Perez Escobar from Kew Gardens, Isa Blasco, which is former PhD student of mine, which is now in the Natural History Museum of Geneva. Cristina Llopis, also former PhD student of mine, which will come soon back to Valencia with Margarita Salas, yes? I always confuse all the names. Yeah, Margarita Salas, likely. And my co-workers in the computational biology lab. Something happened with this, but okay. Yeah. Many thanks for your attention.
Well, thank you very much for this very interesting talk. Do we have any questions in the audience?
Thank you very much. It's a very nice talk. It seems to me that you need a lot of data to this computer stuff, and you have to have these congruent histories that are based on something that you already see, some patterns that you already know. My question is, if you don't have these patterns, you can have new information from your computers to make a prediction, for example, not only to make a description of all the stuff that you already know that is congruent with the story, or you can have predictions about how can one, how could be the coevolution between two species, for example. I don't know if I was clear.
Yeah. I think, well, actually, I think you can, because I think that the question here is, because actually there is already a R package, which is called Tree Duncan, which was published some 2 to 3 years ago, that what actually does is to simulate this kind of this kind of problems. So you give them a series of parameters, and then it starts to work and just end up with this sort of thing. So I think this could be used for this kind of work. So because if I recall well, you have a time, so you can stop at some particular time. So you can decide in the future. So you can compare one time to another time. The problem always is how reliable these simulations are, but in theory could be done. So if you are able to reproduce very exactly what happens in nature, you could be able to do it, yes.
Because at the beginning you show a picture where you have this nice picture with the history of the evolutionary history. Can you put the 1 or 2 slide?
Yeah.
The first one. When you have this continent is separated from this continent and so on. So I am wondering if you can do that. I mean,
You mean this thing?
So you can go there, so you can put all these variables to see how was the evolution, the coevolution from Australia separates from Antarctica to here. So this is something like you can do?
Okay, so you mean if you have this particular, this problem, just trying to explain what's happened, you need the historic, I mean the geographical data, but of course you can somehow compare with.
You need information.
Yeah, you need additional information. Yes.
Thank you. Any more questions?
Thank you for this super interesting talk. I have plenty of questions actually, but I will try just to be like just ask a couple of them. So one is a philosophical one. So what is your opinion how these congruent cophylogenies can be? Are they because hosts and parasites have ancient relationships, or because they are virulent parasites, so there is like a selection of hosts, and at the end it turns out in a cophylogeny and a coevolutionary history? Is it a matter of time or a matter of virulence, or what's your opinion?
A matter of what you said?
Virulence, like virulence?
Oh, virulence.
Yeah.
Okay. Probably both.
Yeah, could be both, right?
Yeah, I think what I think is that not all, when we talk about parasites, we are talking about a whole lot of different organisms that have very little in common, apart from their life strategy of exploiting a host. So I expect to find all kind of mechanisms depending a lot on the type of parasite, if the parasite is virulent, if not is virulent, this sort of thing, because this, in a way, also explains why, for instance, Fahrenheit was so convinced about the idea of phylogenetic tracking of their host, because he purposely was studying lice, and these patterns you see here in this slide are very common in lice. But if you look at monogeneans, for instance, the pattern is totally different. So what I would say is that it would depend a lot on the type of parasites and the type of host, of course, that you're studying.
Okay, thank you. I have, if I have the chance, another question. So in terms of the technical part of the PACO package, is also facilitation something that can be like a facilitation that, like in the event of one parasite facilitating the invasion by a second parasite, can be like controlled or understood by the results of the PACO, or I don't know if this makes sense, this question.
Actually, it's a new idea you come up with. Didn't think of before. Therefore, I would say no, but something that is worth exploring.
Okay, thank you.
I have a question related to that. Can you handle multiple parasites at a time with these methods?
At the same time,
I mean, host versus couple of parasites?
2, 3, 4, yeah. Well, there is a method published that never came very popular. I remember there is a paper where they use a tripartite relationship, so they have one host and 2 parasites. The problem here is that you have to consider, I mean, the problem increases exponentially when you incorporate more. So, because there will be interactions as well. Not here you have, these guys are related to these guys, but if you introduce a second parasite, so in addition to looking at this host with these parasites, you have to also look at the interaction of these parasites with these parasites. And therefore, from a technical point of view, it's more difficult to deal with. But right. Again, it would be very interesting because you have problems like the one of the fig pollinators, where you have 3 different types of insects that are found in the figs. So,
yeah, as Rodrigo was saying, some parasites might facilitate the appearance of further parasites.
Thank you very much for your talk. We have seen many examples about the symbiosis between 2 metazoa. Have you tried to do the same analysis for analyzing the symbiosis between a metazoa and an unicellular organism like bacteria or small eukaryotes?
Well, you can apply these methods to any kind of organisms.
Yes. Okay.
Basically, I think what can give you more insight, apart from, of course, doing the comparison, is, and here is where the things get complicated, is when if you apply the Cophylogeny space, what to include here, okay? Trade distances. Yeah, because we have published a couple of things, And for demonstration purposes, it works, okay? Because here we use the anchors that are very important for attachment to the host. So we think that this is important, explains is adaptive, is very adaptive, ecology and so on. So it would explain a lot of the relationship. But in the case of the fish, we just used morphometrics of the fish. And we acknowledge that it's not the best traits to use, but for demonstration purposes, it works, okay? So, but the idea is if you have a metazoan and you have a unicellular organism, and you want to apply this sort of thing, then the key question here is if you have which traits to place here, and if you have information of these traits.
Okay, thank you very much.
Okay, is there any quick last question?
So I guess this just considers whether, in this case, a parasite can infect the host, right? But some parasites may be better at infecting some hosts, and other hosts, they don't infect all that well, but they can still be found. Is there a way of incorporating that, like their ability for infection, or just whether?
Okay, that's one of the things in the to-do list, because here you see, by now interactions are coded as present absence. So by now, but I think it's possible to somehow, because in bipartite networks, you have weighted networks where the links don't value the same, because it depends on the intensity of the relationship, and this quantitative information should be incorporated in the. I think the reason why we are just working with presence absence is because initially the studies were at a very microecological scale. So you were more interested in you have a large phylogeny of this, how to see how they diversify together or something like that. But if you just go to a more local or regional or whatever study, then I think you need this and should be incorporated. But by now, not. But I don't think it would be very difficult to. I think somehow just adding, when you do for instance, the sum of square residuals in PACO, you could add some kind of weight to each residual that could reflect, and so you end up with M2 that would. The problem there would be with how to normalize this value, but looks feasible.
Okay, so we're gonna stop the discussion here. Thank you again, Antonio.